Every single time you scrape your knee and watch that skin heal, or honestly, every time you grow even a single millimeter, this massive, microscopic factory inside you fires right up. In just a matter of hours, this factory perfectly copies billions of data points, and it does it without you even noticing. It's literally the ultimate manufacturing process, and it's happening right now, all over your body.
But here is the big question. How exactly does a single cell perfectly copy all of that information? We kind of take it for granted today, but for a long time, the mechanics of something on this sheer biological scale were a complete mystery to us. So in today's explainer, we're going straight down to the microscopic factory floor to see exactly how scientists figured out this incredible biological puzzle.
Okay, let's dive into this. Here is our roadmap. First, we'll look at the blueprint of life, then the 3 competing DNA models, followed by solving the DNA mystery. After that, we break down the actual process into 3 steps: initiation, unzipping the genes; elongation, building new strands; and finally, termination, finishing the process. All right, jumping right into section 1, the blueprint of life and the basics of biological inheritance. So before any cell can divide into 2, it has one absolute prerequisite. It literally must duplicate its DNA. If it doesn't do that, there's just no way to pass the genetic information on to the new daughter cells.
And this entire process happens through a foundational concept we call semi-conservative replication. Simply put, this means the original DNA molecule, which, as you know, is made of 2 strands, splits apart. Then each of those original strands serves as a super precise template to produce a brand new complementary strand. This exact mechanism is the whole basis for biological inheritance across all living organisms. But here's the catch. We didn't always know it worked this way.
Which perfectly brings us to section 2. 3 competing DNA models, and the great scientific puzzle of the 1950s. You see, after Watson and Crick famously proposed the double helix model for DNA, the scientific community hit a massive roadblock. They knew the shape of DNA, right? But they had absolutely no idea how it actually copied itself.
Now, what's really interesting here is how visually distinct the 3 competing theories of the time were. Look closely at the parent strands in purple and the newly built daughter strands in light blue. The conservative model up top suggested the original DNA somehow stayed completely intact, just magically generating an entirely new double helix. Then, the semi-conservative model in the middle argued that the strands actually split, meaning each new molecule would have one old purple strand and one new blue strand. And finally, the dispersive model on the bottom? That one proposed a messy, interspersed patchwork of old and new DNA along both strands.
So, which one was it? Let's uncover that in section 3, solving the DNA mystery. In 1958, 2 truly brilliant scientists, Matthew Meselson and Franklin Stahl, decided to crack this genetic cold case once and for all.
And let me tell you, their methodology was so incredibly elegant that it went down in history. People in the field widely revere it as the most beautiful experiment in biology. This was definitely not your run-of-the-mill lab test. It was top-tier scientific detective work.
Here is exactly what they did. They essentially color-coded the DNA using nitrogen. Step 1. They grew bacteria in a heavy nitrogen, known as N15, forcing it to become part of the bacteria's genetic code. Step 2. They moved those same bacteria into a light nitrogen, N14. That meant any brand new DNA the bacteria synthesized would have to be light. Then they simply let the bacteria replicate and used a centrifuge to weigh the DNA after 1 round and then again after a 2nd round of replication.
And here is where it gets brilliant. Just look at these test tubes. After that first replication in the light nitrogen, the centrifuge showed just one single intermediate band. It was hybrid DNA, not fully heavy, not fully light. That finding immediately completely destroyed the conservative model, because that model would have shown 2 totally distinct bands. Then, look down at the bottom tube after the 2nd replication. We see 2 distinct bands now, one hybrid and one completely light. That single image right there flawlessly proved the semi-conservative model and completely ruled out that dispersive patchwork theory. The mystery was officially solved.
Okay, so now that we know how it works in theory, let's check out the actual mechanics in section 4, initiation, unzipping the genes. This is where we officially start the cellular factory.
I want you to think of this process as a highly orchestrated microscopic factory floor. And here is our active roster of construction workers. First up is helicase, whose sole job is to aggressively unzip the DNA double helix. Then we have RNA primase, kind of like the foreman, who drops down a flag to say, hey, start building right here. Next is DNA polymerase, our main builder, synthesizing the new strands. And finally, ligase, the finishing worker who glues any remaining gaps together.
But here's the thing, the factory doesn't just open up randomly. It always starts at very specific locations called the origins of replication. These origins are uniquely packed with adenine and thymine sequences, or A and T. Why? Well, because A and T are only held together by 2 hydrogen bonds, while C and G bases have 3. So it simply takes way less energy for our unzipping worker helicase to use ATP and pry apart those double bonds to swing the factory doors open.
And this brilliantly illustrates what that unzipping actually looks like across different forms of life. Over on the left, we've got a prokaryote. Think of our E. coli bacteria. It has just one single circular chromosome, so it opens a single origin point, creating one replication bubble that expands in both directions. But on the right, look at the eukaryotic DNA. That's the kind you and I have. Because our chromosomes are so massive and linear, we actually have multiple origins of replication firing off all at once, creating several bubbles that eventually just merge together.
Once those factory doors are open, the real heavy lifting begins in Section 5, elongation, building new strands. Now, when helicase unzips that DNA, the 2 strands naturally just want to snap right back together. To stop that from happening, special workers called single-strand binding proteins basically act as wedges to prop the 2 strands open.
Now, we absolutely need to talk about the golden rule of this factory floor.
Our main builder, DNA polymerase, is incredibly fast and highly accurate, but it's a bit of a diva. It has a massive limitation. It can only work in one direction, from the 5' end to the 3' end. Plus, it flat out refuses to build from scratch. It requires an existing free 3' hydroxyl group just to attach the very first piece. Because of this extremely strict rule, RNA primase has to jump in first to lay down a short, temporary RNA primer. That gives our diva DNA polymerase the exact starting point it demands.
To see why this rule causes absolute chaos on the floor, look at this anti-parallel diagram. DNA is anti-parallel, meaning one strand runs 5 to 3, but the opposite strand runs 3 to 5. Because our replication fork is opening up in one direction, but our builder can only lay down tracks in the 5 to 3 direction, it literally forces our molecular factory to work in 2 completely different ways at the exact same time.
So we end up with a leading strand and a lagging strand. The leading strand is an absolute dream. DNA polymerase just latches on and smoothly zips along continuously in the 5 to 3 direction, perfectly following the fork as it opens. But the lagging strand? It's facing the completely wrong way. It has to be awkwardly built backwards in these short, fragmented chunks. That means RNA primase has to frantically lay down new starting lines over and over again. And worse, all those temporary RNA primers later have to be painstakingly removed and replaced with actual DNA.
This totally frantic building process just races along until we finally hit Section 6, termination, finishing the process. Basically, elongation just keeps going until the expanding replication forks finally crash into each other. When they do meet, the factory has to safely shut down. The newly synthesized DNA is left with thousands of tiny little gaps, especially all over that fragmented lagging strand. So, ligase swoops in like microscopic glue, sealing every single final nick to make the strands perfectly smooth and continuous. Oh, and in circular bacterial DNA, the 2 complete daughter chromosomes end up interlocked together like rings. So, a specialized enzyme called topoisomerase IV acts like a pair of molecular scissors to snip them, separate them, and reseal them. And with that, the work is finally done.
The sheer precision of this entire process is honestly staggering. Billions of base pairs, unzipped, primed, copied, and glued together. But it does leave us with one final, pretty provocative thought. What actually happens when this incredibly complex factory, even with all its built-in proofreading systems, inevitably makes a microscopic mistake? Well, as it turns out, those rare, uncorrected errors in the blueprint aren't always a disaster. In fact, those tiny little mistakes are the exact mutations that drive evolution, creating the breathtaking diversity of all life on Earth. Pretty amazing, right?